Hi guys. I've decided that I'm going to do a metabolism review with you today. So we're going to go over some of the finer points of photosynthesis, and then we will go over respiration, and then at the end I will throw some questions straight out of the review packet that you got, the theory review packet that you got, and kind of work them. I'll give you a little bit of time to work them, and then I'll work them and show you where you would have gained points. You probably will need a couple of pieces of just regular plain paper or line paper, whatever. A lot of this is notes that you've had before. I just kind of took our PowerPoints and pulled what I thought was the most important things and the key things that we needed to go over. So if you want to rewrite some stuff down or jot things down, feel free. If you want to kind of follow along in your theory packet, feel free, but we will be using that theory packet closer to the end. This is a huge topic with two major players, so it's probably going to take us the whole time to get through it. If you need to pause so that you can get some supplies, paper or whatever, y'all know where paper is in my room, then somebody go ahead and pause the video and just start me back up when you're ready. Okay, so we're going to start off with photosynthesis first, and just want to remind you of some of the major things. So photosynthesis is divided into our light-dependent reactions and our light-independent reactions. It happens in the chloroplast, which are found in lots of cells that have photosynthetic ability, because this is where photosynthesis obviously happens, so it kind of goes hand in hand. Remember that your light reactions or your light-dependent reactions are going to take place in the thylakoid membranes, which you can see right here, and they are going to make reactants for the Calvin cycle, which happens in the stroma. So the whole point of your light-dependent reactions, as their name suggests, is to take the energy coming in from photons of light, to take water molecules and split them and create electrons, which we can then pass energy through and harness for energy later on in the game. Through that movement of electrons, we are going to create two things. We're going to create ATP, and we are going to create the electron carrier in the form of NADPH. One of the end products, or one of the products of this whole process, is going to be oxygen. Now on the Calvin cycle end of things, we're going to use those products of the light-dependent reactions, again that would be your ATP and your NADPH, as the reactants for the Calvin cycle. And the Calvin cycle is all about taking that energy source from the light-dependent reactions, utilizing carbon dioxide and forming sugars. So we're building up energy-rich molecules here using carbon dioxide and energy. Now there are some major similarities between our photosynthetic reactions or our photosynthesis reactions, and our respiration reactions. And one of those would be the presence of an ETC, or an electron transport chain. It looks very similar to the electron transport chain that you've seen in cellular respiration. It's all about those membrane embedded proteins on highly folded inner membranes, which just increases our surface area. It makes us more efficient. It makes us more productive. When I say us, meaning the organisms capable of utilizing these. Your electron carrier for photosynthesis, there's only one, is going to be in the form of NADPH. So it's all about pumping hydrogens across the inner membrane. One of the reasons that all of these main energy makers are double membraned. And utilizing a specific enzyme called ATP synthase to create some ATP, cellular currency, cellular energy currency. Now remember that these two reactions are kind of the opposites of each other. So the ETC in respiration, while it looks very similar and it pretty much is utilizing the same major kinds of parts and pieces, at the end of the day, for respiration, it's all about breaking that energy source down and releasing ATP, whereas for photosynthesis, it's about taking some energy to make more energy-rich molecules. Another difference is, and kind of similarity all at the same time, is the fact that your respiration reactions are also going to create and utilize electron carriers, but this time in the form of NADH. That's the main one, but there's another one called FADH2, which we will talk about in a little bit. Right, so like I said, for the electron transport chain of photosynthesis, we're going to be utilizing the electron carrier NADPH. That's where we're going to store some energy for later on. And then we're going to be making a little bit of chemical energy in the form of ATP so that we can later on make our glucose molecule. But this is pretty much the arrangement of the ETC within a thylakoid membrane. So remember, those thylakoid membranes look like stacks of pancakes. It's like one pancake and then another and then another. So the arrangement of the molecules is slightly different to the ones that we are familiar with for respiration. But it's pretty much the same concept. We have these huge protein enzyme complexes embedded in the membrane, and we're going to go in and hit it with a photon of light. We will have taken up water via our root system. That water molecule is going to get split. The oxygen is going to be released as a byproduct, and the hydrogens get pumped into the inner membrane space. Now, before those hydrogens get pumped, we actually turn them into protons. Notice the plus charge, because we're going to strip them. We're going to borrow some electrons, one from each hydrogen, and we're going to utilize the photosystem organization that these chloroplasts have to both harness the power or the energy from light, and also to move those electrons around and create ATP as we go. So we hit it with light, the electrons get excited, they move across these proton or protein complexes, and as they move, they lose energy. They go kind of what we call a downhill reaction. And then when we get to our second photosystem, we kind of repeat the process all over again, and we hit it with some more light, and they get excited once more, and then they start moving from complex to complex. Now, at the end of the complex, we're going to go ahead and hand over those hydrogens to NADPH, our electron carrier, and it's going to go ahead and move into the stroma and go to the Calvin cycle to do its role there. Now, these hydrogen protons that we've been pumping into this inner membrane space are going to build up. And we know that things don't like to be on one side or the other side. They kind of like to be even out. So they're going to move via chemiosmosis. We pump them through this specific enzyme called ATP synthase. And as they move through ATP synthase, they create ATP. So that's your ETC for photosynthesis in a nutshell. Right, now let's talk a little bit about the pigments that make up these complexes, these photosystems within our thylakoid membrane. So it's a cluster of molecules, all of which hold pigments and are capable of absorbing photons of light. And they're found in certain complexes, certain complexes within the thylakoid. Now, the main player in terms of pigments are going to be chlorophyll, but there are others that become important, especially as we change seasons and the kind of light that our plants or our organisms are exposed to changes. So like I said, these are embedded in your thylakoid membrane. They're arranged in clusters, which are called photosystems, and it's just because they're a collection of molecules that all do the same thing. They harness photons of light or energy from light. And it's all about structure and function, as you know. Molecularly, they have the perfect structure for this light absorption that we're going to utilize them for. Okay, quick reminder about our visible spectrum of light. This is important, especially when it comes to these colors and their corresponding wavelengths. Okay, don't forget, the shorter the wavelength, the higher the energy. The longer the wavelength, the lower the energy. Of course, Roy G. Biv, always a good way to help you to remember how to organize those colors. Now, because different wavelengths of light correspond to energy, that's where the energy part of this comes from. So we're literally getting energy by absorbing those wavelengths of light. Chlorophyll A absorbs best in a red to blue-green wavelength, and it absorbs the least in the green section of the spectrum, which is why most of our plants look green. It's because that's where chlorophyll A, which is our predominant pigment, is pretty much reflecting that particular wavelength. So this is a question that you didn't do that great on the mock test. You pretty much got not this exact same graph, but a couple of graphs that looked like this, and it's all about being able to read and interpret those graphs. Guys, do not forget that your axes on your graphs, if they are labeled like here, well, sorry, that didn't work out like I planned, like there, and like there, this is giving you information. This axis, sorry, you can't see what I'm pointing at. This axis right here, hopefully, there you go, is telling you about the absorbance of light, like how much light is being absorbed. Whereas this axis is telling you what's the corresponding wavelength that we are absorbing. Wherever you have peaks, that means that absorption is taking place. Wherever there are very low peaks or no peaks whatsoever, where it dips down really close to that x-axis, there is very little absorption taking place. There is a reflection happening at that point. You have to be able to read these graphs. Just by looking at this, you should be able to tell that this molecule right here, which is chlorophyll A, looks like it's absorbing in this purple region, not really absorbing in this bluish, greenish, yellowish region, and then absorbing again in this orange, reddish region. All of that from the graph. Wherever that peak goes up, based on this label, we're looking at absorbance. Now, these other lines show some of the other pigments that we have. Chlorophyll B is another kind of pigment found in those photosystems. We have carotenoids, we have xanthophylls, a bunch of them, and they absorb in different areas. They all kind of generally absorb in the same area, just smaller wavelengths here and there, or bigger wavelengths here or there. But again, it's the same thing. Wherever you have a peak, that's where absorbance is happening. Wherever it plateaus out or it levels out, it's getting closer to zero, it means that you have reflection or very little absorbance taking place. It's super important that you are able to explain that when you write these free response answers. Okay, so our photosystems are arranged in the opposite order. We have photosystem 2, whoops, sorry, I always do this. We have photosystem 2 right here. We have photosystem 1 right here. They're pretty much just named for the order in which they were found, so in the grand scheme of things, it's not that big a deal. I'm not going to go over this picture because I've already gone over this picture, but this is just to give you it's kind of a bigger or more zoomed in version and up close view of where those photosystems are arranged. All right, so one more time with enthusiasm, we're going to go ahead and get energy in the form of sunlight. And that energy is going to come in. Water, which we've taken up via the root, is going to get split. The oxygen is going to leave via the stomata as a byproduct, and we're going to go ahead and pull off the electrons from the 2 hydrogens of water, and we're going to get them excited using that energy from our light that came in and shone through our leaf. When the electrons get excited, they change states, they literally get energized, and they're going to start moving from complex to complex to complex. As they move, they're going to lose energy. So once they leave photosystem 2, which is where they're first energized, when they get to photosystem 1, they will be lacking energy once more, and they will need to be re-energized via some more sunlight. Once they're re-energized that second time, again, they are passed from complex to complex until they're finally handed over to NADPH, which is our electron carrier for photosynthesis. Now, hydrogens have been continuously being pumped through this membrane and into the intermembrane space where they build up and collect. And as we know, because of the balance of things, sorry guys, they're going to eventually start moving so that they're not just concentrated on one side of the membrane. It just so happens that they're going to move through this enzyme called ATP synthase, which ironically enough makes ATP, and this process is called chemiosmosis. It's the movement of ions across a membrane. Once these ions move, they're going to provide the driving force or the energy needed to couple ADP to inorganic phosphate, thereby creating ATP, and ATP and NADPH both go to the Calvin cycle where we can start making the backbone for glucose molecules. Now, the light-dependent reactions can happen in 2 ways. They can be cyclic, or they can be non-cyclic. When the system ends in the production of both ATP and NADPH, so if you follow this blue line, which you can kind of see, this is going to be non-cyclic photophosphorylation. We go in one way, we come out another way. But if NADP plus isn't available to accept those hydrogens, then they can't just kind of sit there and wait on NADP plus to become available. So instead, the system will cycle back, the system will cycle back, those hydrogens will get pumped back into that intermembrane space, and they will go into the creation of ATP. Okay, so this part, that red part, would be the cyclic photophosphorylation. Okay, so the Calvin cycle, also known as the Calvin-Benson cycle, takes place in the stroma of the chloroplast, and this is where our sugar is going to get made. So we've made the energy necessary to actually couple oxygens and hydrogens and carbons together to make sugar. It happens in this Calvin cycle, and it happens in 3 turns of what's a 3-step process. Step 1 is to take carbon dioxide, which our plant has access to via its stomata, and to use the enzyme Rubisco, which stands for ribulose bisphosphate carboxylase, to remove the carbon out of the carbon dioxide, release the oxygen, and add it to a molecule that's 5 carbons big before the addition of this new carbon. And that molecule is called RUBP, or ribulose bisphosphate. So if you look at this part of our diagram, you can see that our incoming carbons are all red. So 1, 2, and 3. That tells you that we need 3 carbon molecules to make these molecules 6 carbons apiece. Okay? Now, once we have, this step is called carbon fixation because we're literally fixing carbon in place on RUBP, or in ribulose bisphosphate. The next step is to take those molecules and break them in half, split them up into bits of 3. So now you would notice that every other group of 3 has that red carbon. And remember, those red carbons are the ones that we got from, those are the ones we got from carbon dioxide. Let me fix this real quick because now it's crooked. There we go. So after this breaking up step, now we can't just, reach our hands in and break these molecules. We're going to need energy in order to do it. So we're going to utilize 6 ATP, 1 for each new molecule we're making, if you want to think of it that way. And that ATP, comes from the light-dependent reactions because we've already made it. So this is where our ATP that we made in step 1 is being utilized. Now we go through a couple more rearrangements, and then the next step is that we're going to reduce this molecule. Now you know that reduction here is actually a gain, so we're adding on hydrogens to our carbon molecules that we just broke in half. Those hydrogens are going to come from our electron carrier, NADPH, which again, we made in step 1 in those light-dependent reactions. So we add on hydrogen to the carbons, and the result is the building material for glucose, and that would be a molecule that's called glyceraldehyde 3-phosphate, or GTP. Now, once we've gotten our GTP, realize that the GTP has to come from red carbons only. It can't come from these blue carbons. These blue carbons have to be left in the system so that we can make RUBP back. We can put it back into a 5-carbon unit and start this process all over again. If we use any of the blue carbons, then we will be short carbon molecules for RUBP. So that's why we have to input new carbons in the form of carbon dioxide. Okay, so this slide is just to remind you a little bit about G3P, or glyceraldehyde 3-phosphate. It is the end product of the Calvin cycle. So remember, the end product of the Calvin cycle is not glucose, it's G3P. It is an energy-rich 3-carbon sugar, and because all of the organisms that produce this sugar are going to be making carbons or making molecules that are 3 carbons big, we call this C3 photosynthesis. And it's to distinguish between the 2 other forms of photosynthesis, which we will get to in a little bit. G3P is an intermediate, but it's important because it is the building block for pretty much any of the biomolecules. So any of the biomolecules that a living system needs. So let's talk a little bit about Rubisco. So Rubisco is the enzyme that does step 1 of the Calvin cycle, which is carbon fixation. So it takes carbon from the air, literally from carbon dioxide in the air, and it strips the carbon off of the carbon dioxide molecule and attaches it to the 5-carbon molecule of ribulose bisphosphate. It's the most important enzyme in the world because when it fails, it makes life difficult for all of us. And it's definitely the most abundant enzyme because pretty much all green things have it. No, not green frogs or anything, but green photosynthetic things have it. Okay, so let's go over the numbers. When we did this, when we actually learned this process, it was difficult then, but you need to see it again. So the accounting is slightly complicated. It takes 3 turns of the Calvin cycle to make 1 glyceraldehyde 3-phosphate molecule. 3 turns because you only get 1 new carbon from each carbon dioxide molecule. So 3 CO2 molecules give you 1 G3P. So to make a glucose molecule, it'll actually take 6 turns of the Calvin cycle. Because glucose has 6 carbons, again, you only get 1 new carbon from each carbon dioxide molecule, so you need the Calvin cycle to happen 6 times, to turn 6 times. So 6 CO2s give us 1 C6H12O6, or glucose. All of this, in terms of energy, for each glucose molecule, it takes approximately 18 molecules of ATP and 12 molecules of NADPH. Any ATP that's left over, the cell is going to use someplace else. It may not use it for photosynthesis, it'll use it for something else. Energy is always welcomed. Now let's go back to Rubisco. While it's a great enzyme, there are some limitations, and the limitations happen when we look at the changes in the amount of carbon dioxide present versus the amount of oxygen present. Now Rubisco is in the Calvin cycle. It's our carbon fixation enzyme, and its job is to bond carbon from carbon dioxide to RUBP. Now, it doesn't just work on carbon dioxide, but carbon dioxide is its optimal substrate. Just like how we have optimum temperature, carbon dioxide is the very best substrate for Rubisco to get carbon into the Calvin cycle. And the reason we need that is because it's about building sugars and reducing ribulose bisphosphate, breaking it down into pieces so that we can have those carbons we need. Now, when carbon dioxide levels are high, there's no issue. Photosynthesis happens exactly as planned, exactly as I've explained to you, exactly as the book tells you. But when carbon dioxide levels are lower than oxygen levels, so when oxygen concentration is high, Rubisco is going to bind to oxygen over CO2. So oxygen becomes a competitive substrate for the enzyme Rubisco. It's going to compete against CO2 for the active site of that enzyme. Well, that's not as good of a situation. When Rubisco binds oxygen instead of carbon dioxide, that process is called photorespiration, and it leads in the opposite things happening. Instead of reducing RUBP, we're going to oxidize RUBP, and instead of building sugars, we're going to break sugars down. So we're not making glucose, instead we will be utilizing glucose, which is not what we want. Right, so here's just a little mock-up of what it looks like when Calvin cycle has to happen when oxygen levels are high. When Rubisco binds oxygen, I forget that it defaults to the select tool. When Rubisco binds oxygen, we're still going to have a 5-carbon compound. We're still going to split that 5-carbon compound, but instead of getting 2 3-carbon compounds, we're going to get a single 3-carbon compound and a single 2-carbon compound. That 2-carbon compound gets lost as CO2 to the mitochondria, but we haven't made any ATP and we haven't made any sugar. So it pretty much stops the process of the Calvin cycle right at this step. Okay, so the major thing for this is if Rubisco is binding oxygen over carbon dioxide, it is going to short circuit our Calvin cycle. We're going to lose carbons. They're not going to get fixed by the Calvin cycle. We're not going to make any ATP, and we're not going to make any glucose. Okay, so the plant pretty much cuts its efficiency, or the organism pretty much cuts its efficiency in half. What does this mean? Well, it means that in certain environments when oxygen levels are high, plants and other photosynthetic organisms are going to have to find other ways of fixing carbon. So we literally have two other methods of doing photosynthesis that have evolved because of selective pressure from the environment to find an alternative answer. So the alternative is to physically separate where carbon fixation happens from the rest of the Calvin cycle, or to separate when carbon fixation happens. So we can separate physically by changing its location in the cell, or we can separate by the time of day, doing certain things during the day and doing certain things at night. Now remember, the reason that these plants have to come up with an alternative is because oxygen levels are high. Well, why would oxygen levels be high? They'll be high in environmental conditions that are really arid and really hot. So think the desert. Now plants open their stomata so that they can exchange gases. But if it's really hot outside and you pretty much have the windows to your soul open, then you're going to dry out really quickly. So one of the adaptations of plants that live in hot and dry climates and environments would be to keep their stomata closed during the day. Well, the light dependent part of the reaction is still running, which means we're still splitting water, which means we are still producing oxygen. So after a while, the levels of oxygen is going to build up in the leaf, and it's going to compete for the active site of Rubisco, the oxygen is, against CO2. So that's why we had to come up with this alternative to begin with. So again, if we look at C4 plants first, these are going to be plants that do actually two things. Physically, they separate where carbon fixation happens compared to the rest of the cycle. They do it in different cells. And instead of storing your carbon in a three-carbon compound, they store it in a 4-carbon compound, so hence they're called C4 plants. To make sure that they really don't have anything to worry about, instead of using Rubisco, they will instead use an enzyme called PEP carboxylase to fix CO2. PEP carboxylase is the exact same thing that Rubisco does, but it has no affinity whatsoever for oxygen. Now, the alternative version would be CAM photosynthesis. CAM plants are going to separate by the time of day. They're going to fix carbon at night when it's cool outside and it's okay to open their stomata because they're not going to dry out. They're still going to store carbon in a 4-carbon compound, which is a type of acid. That A in CAM stands for acid. And they're going to perform the rest of the Calvin cycle during the day when it doesn't matter if their stomata are opened or not. Okay, so when did Rubisco start becoming an issue? When did C3 not work for everyone? Well, it's possible evolutionary baggage, because Rubisco would have evolved when plants first started evolving themselves and first got their photosynthetic abilities. And that would have happened at a time when in Earth's atmosphere, carbon dioxide was extremely high because we didn't have oxygen, at least not a whole lot of oxygen, until plants became a contender in terms of our environment. So back then it wasn't a big deal because there was so much more carbon dioxide than oxygen. Rubisco never really had to deal with it. But now that our oxygen levels are so much higher, think 21% versus 0.03%, it's a very different environmental condition. We have more oxygen, which means that photorespiration can definitely happen, especially in situations where the plants are exposed to really high temperatures, really dry conditions. All right, so at this point we are done with photosynthesis. If you need to pause and take a quick break, run to the bathroom, stretch, whatever, please try to limit this to about 5 minutes because we still have to get through respiration and then we still have questions that we need to look at. So pause if you need a break. All right, so here is our mock-up of cellular respiration. Quick overview. You know that it happens in 3/4 steps, depending on if you consider pyruvate oxidation a step or not. You know that one part at least happens in the cytosol, while the rest of it takes place in the mitochondria. And hopefully you remember that cellular respiration is all about making ATP. That ATP production happens in two ways or at two levels. There is substrate level phosphorylation, which makes ATP, and there is also oxidative phosphorylation, which makes ATP. The steps individually are glycolysis, Krebs, and the electron transport chain, and oxidative phosphorylation. Those are the major ones. And then there is a step that happens right around here that's not showcased by this diagram, which is called pyruvate oxidation. It doesn't necessarily make ATP, but it does make electron carriers, which we will talk about in a little bit. Okay, so the mitochondria makes a good place for cellular respiration, kind of the same way that the chloroplast makes a good place for photosynthesis. It has this highly folded inner membrane, which allows us to increase our efficiency. There are 4 metabolic stages. One stage is anaerobic, so without oxygen, and the other 3 are aerobic, so in the presence of oxygen. The anaerobic stage is glycolysis, where we're doing respiration, but we're not in the presence of oxygen, and this happens in the cytosol, the liquid part of your cell. The aerobic processes all happen in the presence and using oxygen, and it happens in the actual mitochondria itself. And those would be pyruvate oxidation, Krebs cycle, and the electron transport chain. Right, so when we break down the word, we're going to start with glycolysis. Glycolysis literally means the splitting of sugar. So we're going to start off with glucose, which is a 6-carbon sugar molecule, and through a series of 10-stepped individually enzymed reactions, we're going to break it down into 2 molecules of pyruvate, and those pyruvate molecules are 3 carbons big. This is an old pathway used to produce energy. It doesn't produce a lot of energy, but it's the starting point of all cellular respiration. Glycolysis didn't happen. The rest of what we are about to talk about could not take place. Like I said, not efficient. Only creates 2 ATP molecules for every glucose. So that's definitely not enough for large living systems, sometimes even small living systems, to work with. Okay, so like I keep telling you, nature has this whole if it's not broken, don't fix it approach. The enzymes for glycolysis are well conserved. Nearly everything that does, everything that does cellular respiration starts off using the same parts and pieces, which means that the instructions for making these parts and pieces are going to be in our DNA. This process is so old because it first started in prokaryotes. Remember, these are our cells with no major organelles. There's no mitochondria to do fancy schmancy cellular respiration. We have to rely on good old glycolysis happening in the cytosol to produce just enough energy that we can live. Remember that at the time these organisms are evolving, Earth does not have a whole lot of free oxygen. These guys were around way before plants were around. So the amount of oxygen available, very, very short demand, very small supply. So they had to be able to harness energy and to utilize energy from what the foods that they were eating or from organic molecules available to them without the presence of oxygen. Okay, let's do a quick run through of what glycolysis is. We're converting a 6-carbon molecule to two 3-carbon molecules. That 6-carbon molecule is called glucose. Those 2 3-carbon molecules are called pyruvate, also known as pyruvic acid if we're using old names versus new names. The whole process produces 4 ATP molecules and 2 NAD H molecules. The ATP, of course, is the energy. The NADH is the electron carrier that's going to become important for the electron transport chain. Now, even though we produce 4 total, we're actually only producing 2. Think of it as net pay versus gross pay. And it's because we kind of need, have you ever heard, if you've heard the saying, you need to spend money to earn money, it's kind of the same thing here. You need to burn energy to make more energy. So we need some ATP in order to make more ATP. Really what we need it to do is to make a molecule that's still capable of being broken down and harnessed for energy. So we use 2 ATP molecules, and the result would be that we're giving 2 ATP molecules from the system, or we're getting 2 ATP molecules from the system and 2 NADH molecules. All right, so quick summary again. This is going to be an endergonic process, at least part of it is, because we're going to have to invest some ATP. So when we first start off with glucose, we're doing a bunch of rearranging, we're moving that molecule around, we're breaking bonds, we're going to need energy to do that. So we're going to utilize 2 ATP, and then after those steps have been met, when we go through the second part of these reactions, we're going to produce 4 ATP, and we're going to produce 2 NADPH, and most importantly, we're going to produce 2 molecules of pyruvate. So overall, 1 glucose molecule gives you 2 pyruvates and 2 waters. We lose the waters along the way. We're going to get 2 molecules of ATP and 2 molecules of NADPH, because we have to pay back the 2 we borrow. It's kind of like a loan shark situation. You borrow the money, you got to pay it back unless you want to swim with the fishes. Now, the ATP that's being produced at this level is happening through substrate level phosphorylation. And it's one of the very last steps of glycolysis where we're actually making these ATP molecules. So we're going to utilize an enzyme, and we're going to utilize ADP and a phosphate group from a sugar substrate, which is called P or PEP. We're going to break off that extra phosphate from PEP, which is what this picture right here is showing you. We're going to add it to ADP, and that's how we're getting our ATP and our pyruvate. Okay, so this is substrate level phosphorylation. It's substrate level because these guys are the substrates of this enzyme. So the next step in the process is going to depend on if you are capable of doing respiration in the presence of oxygen, or if there's oxygen available, or if there's oxygen isn't available. If oxygen is available, we're going to go ahead with pyruvate oxidation and Krebs cycle, and we will recycle our NAD plus molecules so that they can pick up more hydrogens and kind of keep that system going. If you don't have oxygen and you are capable, you can then continue respiration in the form of anaerobic respiration, which we call fermentation. And there are 2 kinds. There's lactic acid fermentation and there is alcohol fermentation. Which one you do just depends on, is there oxygen or isn't there oxygen? Kind of depends on who you are. What are you capable of? So alcohol fermentation, we're going to take we've made our pyruvates, we have our electron carriers, we're going to offload those electrons, and we're going to produce some ethanol, and we're going to produce some carbon dioxide. It's a dead-end process though, because we're producing alcohol, and greater than 12% ethanol is going to kill a lot of your prokaryotes. They can't survive in those conditions. It's literally toxic to them, and they also can't reverse the process of the hydrogen transfers. So pretty much dead end, not a good thing. Now some fungi and animals are capable of a different kind of fermentation, which is called lactic acid fermentation. You can go kind of back and forth between pyruvate and lactic acid as long as carbons are available. So this process is reversible. Like once oxygen is available once more, we're going to take that lactic acid or lactate and we're going to convert it back to pyruvate. So this is not a dead-end process. Think of it almost like a long pause button. Okay, oxygen wasn't here, we're going to hit pause and get rid of these hydrogen so that we can have our electron carriers continue to work in glycolysis. And then once oxygen is available, we're going to go ahead and reconvert back into pyruvates and go back into aerobic respiration. So if oxygen is present, the next step would be pyruvate oxidation, or the oxidation of pyruvate. And we're going to transition out of the cytosol and into the mitochondria at this point. So pyruvate is going to enter the mitochondria. Remember, this is going to happen twice, once for every pyruvate molecule, and we have 2 of them. So pyruvate is going to go ahead and enter the mitochondrial matrix. Oxidation is going to take place. So we're going to break it down until we are left with a molecule that's 2 carbons big called acetyl coenzyme A, and 1 molecule of carbon dioxide. In the process, we're going to make some more NADH. So we release 2 carbons for every glucose, because remember, every glucose gives us 2 pyruvates. Each pyruvate is going to give us 1 carbon dioxide. So we release 2 carbon dioxides, and we're going to make 2 sets of NADH, and we're going to make 2 acetyl coenzyme A's. Now it's acetyl coenzyme A that's going to enter your Krebs cycle. And remember, acetyl coenzyme A has 2 carbons. It's 2 carbons big. All right, so here's another schematic of it. Pretty much the same thing the last slide showed. It's just you can see the molecular formulas here and here. Okay, so the Krebs cycle, also known as the citric acid cycle, should look very familiar. It looks a whole lot like the Calvin cycle. Same kind of turning situation. We're still like losing parts and pieces. And we're utilizing carbon molecules going in. So when carbon goes in, it's going to join a 4-carbon compound to make it a 6-carbon compound. And then pretty much all that's happening from this point on is we are getting rid of the carbons we've just put in. We're getting rid of them in the form of carbon dioxide. So right here and right here. We will make some NADHs as we go. We'll also make a little bit of ATP and a little bit of FADH2. And this process repeats twice for every glucose molecule because we're getting 2 acetyl-CoAs coming in because we had 2 pyruvates going into pyruvate oxidation. Okay, so unlike glycolysis, whose claim to fame was that it produces a small amount of both ATP and electron carriers, the Krebs cycle actually produces quite a lot of your electron carriers. It still only produces a couple of molecules of ATP, but those electron carriers are major game changers for the electron transport chain. Right, so here let's go through the accounting for the Krebs cycle. So for every pyruvate molecule, remember we're going to have 2 total. So for every pyruvate molecule, we will produce 1 FADH2 and we will produce 4 NADHs. We're going to take that pyruvate from a 3-carbon compound all the way down to 3 individual carbon dioxide molecules, and we'll make 1 molecule of ATP while we do it. But because this happens twice, the net gain of everything would be 2 ATP, so pretty much the same amount of ATP we made in glycolysis, but this time we're making 8 NADHs and 2 FADH2s. So I get it. So far it's been sounding pretty unglamorous, like none of this is exciting. We're not making a whole lot of anything. But all of that's about to change once we get to the electron transport chain. Now, just like in photosynthesis, there are a series of proteins that are built into the cristae, that inner mitochondrial membrane, and they support proteins and enzymes, and it's all about pumping hydrogens and moving electrons and creating energy. This whole process, and when I say whole, I mean the ETC part of it, can yield about 36 ATP from each glucose molecule. Up until now, we've only been getting about 4, so, 36 sounds really good. The catch, though, is that it's only in the presence of oxygen. So if there's no oxygen, there's no ATC. We're not moving on. You still have to make do with your 4 ATP that you created via glycolysis and Krebs cycle. All right, so you know that once the electron carriers get to this point, they're going to be stripped of their hydrogens. The hydrogens in turn will be stripped of their electrons. The electrons are going to move from complex to complex, and that's going to create the hydrogens, get pumped through those complexes and will build up kind of in the middle section of those two membranes, the intermembrane space or the inner membrane space, where they're going to create a gradient. And eventually, through chemiosmosis, they're going to flow through the enzyme ATP synthase. And that power is going to drive the coupling of ADP to phosphate, creating ATP. This part of the process is oxidative phosphorylation because the attractant for these hydrogen molecules will be oxygen. Okay, so quick reminder about chemiosmosis. It's the diffusion of ions across a membrane. We have built up a proton gradient because we pumped all those hydrogens into the inner membrane space. They are then going to flow through ATP synthase. That is going to drive the coupling of ADP to phosphate. It's chemiosmosis that actually links the electron transport chain to the ATP synthesis part, because they're kind of two separate steps, but they get linked together by that flow of ions. This is where our 36 ATP are being created, by the way. All right, so here is all of it. Starting with, so we know that glycolysis happened in the cytosol. We know that the pyruvates that are made are going to go through pyruvate oxidation. We're going to make acetyl-CoA, which is going to enter the Krebs cycle. The Krebs cycle is going to turn. It's going to produce FADH. Pyruvate oxidation created FADH, and NADH, and glycolysis also created NADH. We're going to create a little bit of ATP, and we're going to create some carbon dioxide. All of those electron carriers are then going to be transported to the ETC. When they get to the ETC, they're going to be stripped of their hydrogens. The hydrogens will be stripped of their electrons. The hydrogen protons will then get pumped into the inner membrane space while the electrons move from complex to complex to complex. And oxygen is going to be the attractive force that is going to make chemiosmosis happen from that inner membrane proton gradient that we've created, which will flow through ATP synthase, thereby providing the power to couple ADP to ATP, making ATP via oxidative phosphorylation. All right, and here's your accounting. You've seen this before. You have this from your notes. We've already talked about this, but it's pretty much just where everything comes from. And at the end of the day, when we couple all of these 4 steps together and everything that they make, we can get approximately 40 ADP per system within this mitochondria. So throughout this whole process, oxygen plays a fairly major role. So what happens if there's no oxygen available at that last step in the ETC and in ATP synthesis? Well, the ETC backs up because there's nothing to attract or to pull the electrons down and create that proton flow or chemiosmosis through ATP synthesis. If the process backs up, it means that NADH and FADH2 can't unload their electrons, or their hydrogen, so they can't go back and collect more hydrogens from the Krebs cycle or from glycolysis. This is going to put a halt to ATP production, and the cell is going to start running on reserve energy, the little bit of energy that is still available. Very quickly, each cell will run out of its energy source, and you will die. This is how cyanide poisoning works. It pretty much binds up the available oxygen and prevents it from becoming that final electron acceptor. All right, guys, so at this point, I'd like you to turn to the questions that go with your metabolism section of your review packet, and I'd like you to work through those. And I don't know if we'll have enough time to go over the answers at the end, but I want you to go ahead and script out, based on the space you have, the best possible answers that you can, and I will see you soon. Goodbye.